Good afternoon, guys. It's Tuesday, and we're going to go over chapter 5. I believe we have it already. Actually, Tuesday morning we had the lecture already, but let's go over this. I'm going to set it up, share the screen with y'all. Make sure this thing is all working. All right, can y'all see that? I hope so. All right, here we go. Got me kind of over here in a little spot here. Okay, so chapter 5 is actually not a really bad chapter here over cell membrane and function. We kind of shouldn't be too terribly difficult to go through. It's going to talk about the structure of the cell membrane and then how we get things in and out of the cell. So we're going to try to roll through this one pretty fast. Got a cool story about the hot sauce there. Y'all can read that sometimes, okay? But if you remember, the cell membrane is a phospholipid bilayer made out of these fatty acids, right? Made out of phospholipids that have two fatty acids, which are long carbon chains, and a charged head is represented with this circle here in the picture that has the glycerol and the phosphate in it. So this end of the phospholipid is water loving, and it forms the outside and the inside of the cell. And then the fatty acids grab each other. They're hydrophobic and they like each other. They don't like water and they grab each other. Okay, then we have cholesterol stuck in here, and this controls the viscosity of the cell membrane. And then we have these different proteins that go in and through the cell. We're going to talk about those quite a bit in a few minutes. So then the components of the cell membrane then would be the lipid component, phospholipid bilayer, proteins sticking in it that serve to carry things in and out, or only on the outside, some only on the inside. So they have function stuff here. And then the cholesterol, which is stuck in the phospholipids to control the viscosity of it. Okay. And we'll look at some of these different things they can do, including there's some sugar stuck on the outside that are your cell markers, your fingerprints of the cell. That tells you, that is your cell only. Okay, and then there are glycoproteins and glycolipids. So here's a more realistic look at it. The model is called the fluid mosaic model because the phospholipids, if you run your hand and get across your arm, they pinch in, kind of like Jell-O. So it's fluid, and these phospholipids can move in and out, so they're flexible. And then the proteins are in a mosaic pattern spread through on top of and under the different components here of it. And we'll talk about those just, but we're going to walk through these different proteins as what they can be. Notice the carbohydrate chains on the outside. So a lot of these are the markers, quote, quote, markers that match you for an organ transplant, for example. And some of these serve as receptors for hormones and other drugs to bind to. Okay, so here's some of the functions of these. I'm going to use the pictures. Some of these proteins are channel proteins, allow things to come in and out of the cell. For example, chlorine, and faulty chlorine channels is the result of that is cystic fibrosis, which is an inherited disorder where the chlorine channel is misfolded and it does not allow chlorine to enter and leave the cell. Pretty bad disease. Others are just carrier proteins, like the real famous one here is sodium potassium pump that pumps sodium out and potassium in using energy, and we've talked about that before, but that's kind of a cool one. Others can be cell recognition proteins. Like I said, the most famous is that these major histocompatibility complexes that are your signature markers that are only person on earth that would have that, like yours, would be an identical twin. So we try to, when we do organ transplants, we try to find someone that has similar markers so we can trick your body into taking that organ. Receptor proteins for hormones and other cell signaling mechanisms. And sometimes it has an enzymatic function inside to catalyze a reaction or so forth. There's another good one there. Cholera bacterial release a toxin that interferes with the proper function of the most famous, probably internal enzyme called adenylate cyclase. And this causes messes up the sodium and water levels. Water leaves the intestinal cell and you end up dying from severe diarrhea, which is a bad way to go. All right, and sometimes it just serves as junctions between cells to anchor cells together, kind of like Legos snapping together there. So that's kind of a cool overview there real quick of these different protein functions. And we'll see some of those showing some multiple choice questions, okay? There's a cool picture here. How does the cell talk to itself? Well, hormones or talk to each other, excuse me. Look at this. Sometimes a chemical like a hormone or a transducing factor of some sort will bind to a receptor, trigger an internal event, maybe the production of a chemical inside that doesn't normally occur, and it could target a structure protein to alter the shape of something. It could make an enzyme to alter a metabolic pathway or cellular function, or it can actually alter the expression of a gene by turning on or turning off genes inside the nucleus. So all those are ways that some of this work, and this is how the human egg develops into the embryo, into the newborn by triggering cells to turn on different genes. That's pretty cool. Now, second part of this chapter is talking about the membrane, the cell membrane being selectively permeable. And what that means is some things can go in, like carbon dioxide, oxygen, glycerol, alcohol, can freely move through the cell membrane, that other things that are charged in large molecules cannot enter and leave the cell easily. Okay? Now, water has special pores called oligopores that allow water to move real quickly, and we'll talk about that. And so we're going to talk about how things can do. And then we're just talking about some physics things here. Active transport is anything going from low to high, with the use of energy in a protein carrier. And then we're going to go look at that in more detail in a minute. That's the way we go against the natural flow. Most things want to go from high to low. Okay, and we'll talk about that. Exocytosis would be the cell getting rid of something really big, chunking out something. Endocytosis would be the cell engulfing a large particle like a bacteria being eaten by a macrophage, one of our immune cells. Okay, and here just showing you how some of these look, once again, where they would be. Sometimes the channels can be turned on and off. There's one showing water, the oligopores, right? But these non-charged molecules, a few can slide in, but big ones can't get through. And so we're talking about these movements here: diffusion, facilitated transport, active transport, and bulk transport. Okay? So diffusion is anything going from high to low. And I go crazy here, dropping some crystal dye in here, and eventually the whole aquarium or the fish bowl here goes to a light shade of purple. So everything wants to go from concentrated areas away from each other. Light molecules want to get away from each other, and that's just one of the laws of physics. Here's the COVID problem. We're having an oxygen transfer problem in the alveoli of the lungs. Oxygen wants to go from high to low into the blood, and this mucous membrane is clogging up, forming a shellac here where the COVID prevents this from occurring by causing inflammation in here that makes mucus that prevents us from getting the gas in. Pretty fascinating there. Okay, osmosis is diffusion of water. Real simple term there. And here's the thistle tube experiment. And notice anytime you do about osmosis, you have to have two solutions separated by a phospholipid bilayer, which in this case is that red. So that's a selectively permeable membrane. Could be a piece of intestinal gut or something in this experiment. And notice that this is 10% solute or sugar and 5% out here. So the water is hypotonic here. The one that has the least amount of dissolved solute is hypo, and the one that has the most is hyper. And water always goes from hypo to hyper. So this will defy gravity and push up and move the level up. And what it's actually trying to do, as that level goes up inside that tube, the number goes down from 10 to 9 to 8. And as it gets concentrated out here, it gets greater than 5. It's going up 6, 7, 8. And so all things try to do is reach this equilibrium. Okay? And we talked about that. If the concentration is equal on both sides of the plasma membrane, it's said to be isotonic. If it's not, then one of them has to be hypotonic to the other solution, and then that solution is hypertonic to it. Okay, so we'll look at a couple examples of this. You can take a cell and put it in an isotonic solution, and water flows in and out equally. If you put it in a hypotonic solution, meaning the water is less concentrated and the cytoplasm is more concentrated, then the water will come in and we'll get the lysis or blowing up of the cell. If you put a cell in a hypertonic solution, meaning there's more dissolved solids out here, that sucks the water out and we get crenation or shrinking. We used to do this with blood. This would be like a cheek cell here, possibly, and here is a plant cell. We do that with a plant cell in lab now using Elidea. And you can see the central vacuole swelling up in a hypotonic solution and shrinking in a hypertonic. But water is always going toward the solution has the most dissolved solutes in it. Okay, facilitated fusion is anything going from high to low with the help of a protein carrier. Now, I know it takes energy to get the protein carrier set up, but after that's going, then no energy is required. So simply these things work their way across till they reach equilibrium. And that's some really bad art. They'll end up with 2 on each side or 3, and then it stops. Active transport takes it a step farther. So we're going to go from low to high and move things against their natural flow. And the sodium potassium pump is one of the most famous ones. It pumps 3 sodiums out and 2 potassium ins as ATP is used to do this. So this requires energy, and that's a big deal. So this is going upstream, shall we say, paddling upstream on a river. Okay. And she's got a nice little setup for showing how that pump works. And I will tell you, I think the art's not too good. But basically 3 sodiums out, 2 potassiums in for every ATP used. Okay. And sometimes we may have to do a big thing. So we have endocytosis. In this case, we're chunking it out, exocytosis. And then sometimes we'll have cellular eating, phagocytosis, or cellular, drinking, pinocytosis, or sometimes chemical binds to a receptor and that triggers endocytosis. And we have an electron microscope showing that there. That's pretty cool. These are just terms you need to know. And then outside the cell surface, this extracellular matrix, a lot of research going into this now, where these collagen, elastic fibers, and others are out there playing a role in cell signaling. Peptidoglycan is a protective layer in the bacterium. So a lot going on out here, and this is the area of a lot of research right here, proteoglycans and all out there, that we're seeing a lot going on out there. That's a hot area of current research. And then there are three junctions that hold cells together. These are three real famous junctions. The first one is called adhesion junction, also called sometimes called a desmosome, right? And then we have tight junctions, and then we have gap junctions, okay? And here is the adhesion junction, the desmosome, and it looks like a spot weld where fibers actually go from cell to cell and help hold it together. It's not waterproof, but it's real tight. It's pretty good. The tightest is the tight junction where plasma proteins from two adjacent cells actually fuse together and form a waterproofing here. So this would be like in the blood vessels or in our gut, so we won't drip acid down into our stomach, or our stomach down to our kidney. That would be bad. So these are real tight, and you get a zipper effect. And then the last one is really kind of cool, is this gap junction where you have two adjacent tunnels from two cells formed together, so you have a hole in there. Ever do it like that? Yeah, so they come together so you have a tunnel in there, and that allows ions and signaling molecules to go from cell to cell very quickly. We see these in cardiac muscle, and this is how they're able to contract in a rhythmic function, all the heart at once. Okay, and then we have the plasmodesmata of the cell wall of plants that allows for material to be transferred between plants. And here's a picture of it. You can see it right there. It's pretty cool. So they have little special holes between their cells. And I think that's it. So it's kind of a short chapter there. I kind of hit that one real quick today in lecture. And we'll stop here and let y'all load that one up. And then we'll come and talk about chapter 6 later. Have a good day. Thank you.